Thank you very much, Professor Adeli, for this very interesting talk. And now we are going to change to other very interesting talk. And as you know, this is a multidisciplinary meeting. So we are going to switch. Instead of talking about engineering, we are going to talk about biology. So it's a great pleasure for me to introduce you Dr. Luis Martinez. Dr. Luis Martinez is working at our university, at the Neuroscience Institute in our university, and he is has been working for many years on the analysis of visual processing. I will say that he knows a lot about the tricks that the visual system is using. And I will also say that Luis is also a magician, so probably he's going to tell us something about what happens in the brain when processing visual information. And I don't know if it is ready. Well, we have a slow computer today. But Luis is going to talk mainly about what happened from the retina to the LGN and to the brain, how the brain is analyzing visual information processing, because I think that it's very important to understand neurobiology and to also to develop new tools, new devices, and is very complementary to the goals of this kind of meeting. Almost there. I hope you have no problem with the movies.
No. Just one of them.
Okay.
Okay. So thank you very much, Eduardo. Thank you for inviting me to discuss my results with this audience, and thank you for bearing with me after this first talk. As Eduardo mentioned, I'm a neuroscientist, and I'm interested in the visual system of the brain, which has to face a daunting task, which is to create in real time an internal representation of the external world. And the challenges that the brain faces in order to achieve this are very common to the challenges that you people in artificial vision have to face or deal with on a daily basis. So, for instance, the brain receives a very large amount of information. I think that the latest estimate, visual information, I think that the latest estimate is something about 70 gigabytes per second, 70 gigabytes per second, which is equivalent to watching 70 different movies at once per second, including images, and musical score, dialogues, everything. So that tells you already a little bit about the strategy that the brain has to use in order to construct a internal visual image of the world. And one of the first things that it has to do is to compress vastly that information and throw away most of it. Okay? This is a problem that is also, the large amounts of information that you have to deal with is a problem that is also common in the artificial world. During this morning session in the world, more pictures will be taken than in the entire product era of analog photography. Just simply in this morning session. That tells you that the challenges that people in this type of business have to deal with in order to sustain communication channels that are able to transmit all this information per second are parallel, very similar to the ones that the brain is facing in order to transmit visual information from the eye to the image processing centers in the brain. Okay. Not only that, I mean, we are very bad at processing certain types of information. For instance, we have no problem interpreting a visual face immediately as soon as we open our eyes. but we're very bad, something that a computer cannot do, and still not do very precisely. However, we have a very hard time at, for instance, calculating the square root of a large number, which is something that a computer can do in a split of a second. I'm just going to give you a hint of how bad we are dealing with certain types of information. So I'm going to do a little trick, okay? I want you to do exactly as I instruct you to do. This is sort of a magic thing, but you're going to actually grasp how hard it is to follow certain threads of information. So all of you, do just this. Put your hand like this in front of you. This. Bear with me. It'll be illuminating at the end. And now get your index finger with the other hand and place it in the small finger of this hand. Okay, you have it. Now, the instructions are going to be to move a number of positions along the different fingers of your hand. Okay, so if I instruct you move 4 positions, you're going to have to move 4 times in the array of fingers of your hand. There's some rules, however. One thing is that you cannot jump positions. You can change directions whenever you want, but as soon as you reach one end, you cannot go directly to the other end. You have to go back. Okay? Those are the 2 main rules. Get it? Please, I can't see you, so just say something. I hear you. Okay, good. So I'm going to try to predict the finger that you are going to end up. Okay? And I'm going to turn around now. I won't see you, so I shout out the instructions, and I say now when you are allowed to move, and then whenever you're finished, just please say so. Say done or whatever. So I realize that we give you the following instruction. Okay? You got it? Good. I'm turning around. Now, you hear me? All of you? Okay. Now move 4 positions. Now. You got it? Done? Okay. Now move 3 positions. Now. Remember, if you reach one end, I'm going to go directly to the other end of change directions. You can change directions whenever you want, but you cannot jump fingers. Okay? Are you ready? Okay. Now move the number of positions that you want to move freely, choose it, but remember the number of positions you move. Okay? Now. Got it? Okay, now repeat the same number of positions moved. Now. Got it? All of you? Yes. Okay, now if you are a female, move 2 positions. To the right. If you are a male, just do nothing. That's it. Now. Got it? Yes. All of you? Okay. Now, all of you, male or female, move 2 positions to the right. Remember to go back to this here. Now. Got it? All of you? Okay. Now, all of you again, move 1 position to the left. Got it? Okay. Now, leave that finger up and take all the others down. And then turn around. Okay? You didn't like it, right? But I hope you like the rest of the talk. This is how hard it is to follow information. I gave you all the information. I even gave conflicting information to different groups of people, but still I reached the same position. Now, if you want to reconstruct the trick, it's not that easy. And this is because our brain is not ready to deal with this type of information. Okay, I'll keep going. Here. Information is also packaged in very funny ways. Here, if I ask you what you see here, most of you would say that a face. It's basically a bunch of trees, and the funny shape of a face emerges from the spatial disposition of the different elements in the trees. This means that we have information in different planes. One is the trees, and the other plane is the face. And the brain has to decide which one is more informative for us and decide on which one are going to influence our behavior from now on. Now, this is easy because both planes are very easy to see at the same time, but here it's not as easy, it's slightly trickier. How many of you can see the arrow? And now raise your hand, the ones that see your arrow. And wait a little bit and more people are going to start seeing the arrow in the picture. Okay, for those of you who cannot see the arrow in the picture, the arrow is actually shaped by the space lived by the letters E and X. Now you see it? All of you? Okay, this is not, I mean, a random feature of our alphabet. This is something that people working at FedEx actually intended to do, simply because they include this illusion in all the signs that they have in different languages and different alphabets. The funny thing is that if I use this alphabet first, the one at the bottom, all of you would have seen the arrow, because the arrow now is the one that carries the information, not the alphabet, unless you speak that language, which I don't even know which one it is. I'm sorry. But because I use the one on top first, the alphabet carries a lot of information for you, and the second plane of information is not obvious at all. So this is something that makes things even trickier for us. Not only that, another problem that the visual system has to deal with is this, which is called the inverse problem of vision. And it stems from the fact that the brain has no access to the real world. All the brain has access to is the projection that 3D objects in the real world cast on our retinas, which is a two-dimensional surface. This is a 3D sculpture by Shigeru Fukuda, a wonderful Japanese artist, highlighting this very same problem. Here you have the white trash in the first plane of the image, that when it's illuminated from a very particular point, which is equivalent to the one you're sitting at, casts a shadow on the flat white screen behind that has nothing to do with the three-dimensional shape of the object that is producing that shadow. This happens all the time, and our brain has to deal with this problem also. We learn to associate 2D projections to 3D objects in the real world based on experience, and we do it statistically. This is a different problem, but this is just to highlight the many problems that the brain has to deal with. Not only that, the thing is that the shadow that natural images cast on the retina are not as crisp as the silhouette of this two person in the background. They are more like this. They look more like this. Can you make out of what kind of 3D object would have to produce this retinal projection? This is simply a projection in 2D of the luminance casted by an image on the retina. Do you know what image casted that projection? Raise your hand if you know. Okay, you've seen the picture before? Okay. This is it. It's a bicycle leaning on a tree. Now that you see the bicycle, then you can actually kind of see where the wheels are, the frame of the bicycle is, and everything. But it's very difficult to predict otherwise, unless you have the real image before. So this is the real problem that the brain has to deal with. And these images are very difficult because they are highly correlated at the local level. So they pose a problem for visual coding. This is a real natural image, which is kind of degraded, and this is the matrix of correlation, pixel to pixel. This is how much the luminance of one pixel correlates to the luminance of its neighbors. So the visual system, what it must do is to efficiently encode this visual information, and it needs to reduce redundancy. Well, this is something that we can discuss at the end if you want, whether reducing the redundancy is a strategy or not to encode visual information, but the dogma goes like this. So you have to reduce redundancy in order to efficiently code information. This is, bear with me with this very light introduction, but I want to set up the points of the analogy between artificial vision and how the brain actually deals with these problems. So this is one way of dealing with this reduction. of redundancy in natural images. Imagine that you have this matrix of luminance test in which the range of the lowest luminance, from the lowest luminance to the largest luminance is very narrow. And this is actually within the range of the natural images. So you have a minimum of 60 and maximum of 120 on a scale that goes from 0 in pure black to 255 in white. One way to do it is histogram equalization, in which you build a new luminance matrix in which you make artificially the darkest pixel 0 and the lightest pixel 255, and you actually change the luminance of the other pixels accordingly, and you end up with an image in which the features are actually much more stable and easier to grasp. When you do that with natural images, like this image, below you have the histogram of luminance distributions, and when you equalize it, you end up with halos, dark spots in the image. It doesn't look very nice, actually. So there are other tricks that you have to do in order to deal with this. So how does the visual system actually do it? Because it's facing the exact same problem. Large amounts of information on the one hand, and on the other hand, the high redundancy in natural images that you have to code efficiently. So how do we go about it? Well, the first thing that you have to know is that the visual system has a compromise to solve between having the best sensor possible in the camera that it uses, and the metabolic cost that it would take to send all this high-resolution information from the eye to the brain. So we in fact have a very high-resolution sensor at the front end of our visual system. The photoreceptor layer of the retina, which is the one that you have here on the left, has 100 megapixels, 100 million roughly of photoreceptor cells tied the outer layer of the retina. Now, if we want to send that much information to the brain, we would need an optic nerve of 100 million cables, which would take up roughly half of the space of each one of your hemispheres. That's huge, because then you would have to upscale the number of elements receiving that information and giving that information not only at the first nuclei that receives visual information, which is the end of the thalamus, but also in subsequent stations of visual processing along the brain, which already take up about half of your entire cerebral cortex. So if you scale up all this structure according to the size of the new optic nerve, your brain would be the size of a quarter of this lecture hall, basically. That's amazing, and that is obviously biologically impossible. So the solution that nature actually came up with is to compress that information. And the compression is extremely efficient because it goes from 100 megapixels at the front end of your camera to just a single megapixel in the output layer. It's the output of the retinal ganglion cells that you have there, the RGCs there, which send only 1 million cables to the lateral geniculate nucleus of the brain, the structure in the middle, which is the one that receives visual information for the first time. In the lateral geniculate nucleus, the information is upscaled again, is expanded again. We have many more units in the lateral geniculate nucleus than in the retinal ganglion, than there are retinal ganglion cells in the eye. And these units are actually segregated in two types. One are excitatory neurons, or the ones that are in charge of having that information moving to the cortex. And then there are the more numerous units that are colored in red. And then you have the blue units, which are very fewer than the retinal ganglion cells in the retina as well, which are interneurons. And these are local processing units, which we'll see later on in the talk, what their role is in processing and transmitting information from the eye to the primary visual cortex, to the brain. Yeah. So the interesting question is, how do you wire this up? And in the first leg of this compressive sensing circuit, the one that goes from photoreceptors to the retinal ganglion cells, is deal with convergence. Convergence that is massive in parts of the retina and not as massive in other parts, particularly near the fovea, or the place where you have the highest acuity. But we have a fair amount of convergence in which different types of information are multiplexed in the same units. But I'm not going to deal with that part now. What I'm going to deal is with the algorithm that actually decompresses the information at the receiving end. The first nuclei in the brain that is dealing with that visual information, the LGR. Okay, let's think about the retina. The retina, the output of the retina comprises two main types of units. They are called the on-center cells and the off-center cells. I code here them in blue and in red, and they are in charge of processing light decrements, the off-center cells, which means that they will fire within their receptive field, the part of the visual field that they analyze. There's a transition from highly lit or prismally lit background to a dark feature. And then the on-center cells, which have the complementary type of response, they code for light increments, and they like situations in which you go from a dark background to a bright detail. Okay? These units are actually tiling the retina in a very regular fashion, like a pixel sensor of a digital camera, if you will. You have here two different mosaics, one for the off-center cells in blue, one for the on-center cells in red. And what you notice is that they are roughly regular in terms that the packing is almost hexagonal. It has some noise towards the tetragonal packing, but it's roughly hexagonal. And then the distance between same-sign neighbors is also rather homogeneous in terms of different points of the retina. They roughly occupy homogeneously the retina. Now, the two mosaics are independent, which means that knowing where one mosaic has its elements, you know nothing about the other mosaic. But this makes that the opposite sign neighbors are closer together than the same sign neighbors all the time. Okay, and they cannot occupy the exact same positions within the mosaic. Now, this was used to make a very strong prediction about one feature of our visual system, which is extremely important, which is visual acuity, the smallest detail in a visual image that we are able to discriminate. So the distance between same neighbors, the Nyquist limit of that distance was thought to actually set that limit. Imagine that you have a single point in space. This point will project to the retina in roughly a Gaussian manner, what is called a point spread function. Now you have 2 points. You will end up with 2-point spread functions. If their separation is large enough, but they are closer together, then their point spread functions will overlap, and you will actually not distinguish them as two different stimuli, but as just as a single one. Or the distance at which this happens is related to the Nyquist limit between this between the separation of the sensors of the same study. So, fair enough. This is the explanation for visual acuity. Good. No surprises. The thing is that when careful experiments were done, particularly in animals, trying to correlate the distance between the sensors in the retina and visual acuity, something failed. And this is a very tricky experiment in cats. Cats are very difficult to train to do any behavioral task. And that's why this was such an amazing fit to be able to do this experiment, in which they train animals from the very beginning of their lives, from the very moment they open their eyes, to recognize the orientation of that grating. And as soon as they recognize the particular orientation, let's say an oblique orientation in this case, they have to jump to the platform that is on the right, and they get a reward in the form of a particular food that they really like. The funny thing is that, well, the experiment one, they would change the spatial frequency of the grating, and as soon as the animals were not able to distinguish the orientation, the particular orientation of the grating, that was the limit of visual acuity. The funny thing was that the spatial frequency of the grating that they were able to discriminate and had nothing to do with spacing of the sensor, which was actually much finer than what the distance of the sensors would have predicted. Then the theory that the same authors came up with was that, okay, maybe it's not the distance between same sign neighbors what actually matters in order to discriminate these gradients, but it's the distance between opposite sign neighbors. And in that case, actually, the match was perfect, basically. This distance actually correlated very nicely with the visual acuity of cats. The funny thing is that these two types of units never collaborate in a way that would have allowed you to discriminate that grading in the brain. So other theories actually, well, this was called hyperacuity because it's a phenomenon that cannot be explained based on the nucleus limit of your sensor. That's why it was called hyperacuity. Hyperacuity is also present in humans, by the way. It's present in all animals that it has been tested. And another theory is actually had to do with the emergence of feature detectors in the brain. Okay, so people said, fine. So the neural correlates of hyperacuity cannot be found in the retina, then there must be found somewhere else. This is just a very famous and very hard to interpret the scheme of the visual system of the brain. And on the very top, on the very bottom, you have the retina. Retina sends information to the lateral geniculate nucleus of the thalamus, and then the thalamus sends information to the primary visual cortex, or V1, right there. And then V1 actually funnels all that information to the 37 other areas in the brain that deal in one way or another with processing visual images. So people just said, okay, if it's not in the retina where we have to look for the neural correlates of visual acuity, then it's got to be in the primary visual cortex. And the reason for that is because the thalamus and the retina basically have the same exact type of receptive fields. And this is something that, oops. Okay. That you see here. The bottom you have the on-center with off-surround receptive field of a retinal ganglion. In the middle, the same receptive field of a synaptically connected cell in the lateral geniculate nucleus, which are very similar, and then another synaptically connected to the LDN unit in the cortex, which shows a receptive field that also comprises on and off regions, but they are aligned very differently. They are now elongated and sitting next to each other. This is a feature detector, it detects orientation in the visual image, and it was suggested that is the particular special arrangement of these orientations, feature detectors in the cortex, would underlies our ability to discriminate the detail in a visual image beyond the limit of our sensor. Okay, but the funny thing is that the circuit that links the retina to the LGN, even though it produces receptive fields that are very similar, can transform visual information in a very profound way because of the different numbers of units that you have in the LGN and in the output of the retina, right? So the question of how do you wire these elements up is extremely important in order to know exactly what the role of the lateral geniculate nucleus is in processing information. Is it a simple relay that takes the retinal output and transform and send it to the cortex, or it does something meaningful with it that actually adds value to the visual information on its way to the primary visual cortex? Okay, in the thalamus you have the same elements, the same units. You have on-center cells and off-center cells that process information in a very similar way to what I have just described for the retina. Now, this is a recording. We do these experiments in cats as well, so everything that you're going to hear about comes from cat, but it's analogous to what you would find in the human that you can discuss later. The numbers change, but visual acuity changes, different. But the algorithm is likely to be the same. Okay, these units we recorded intracellularly, which means that we have access to the inputs of these cells, to the retinal inputs. We know exactly what's the compound effect of all the retinal input to each one of these neurons. And this is what you're going to see in this slide. Here you have on top the three-dimensional, well, in this case it's in 2D, but in real data is in 3D reconstruction of one of these units. So we know everything about it. We know where it was sitting in the LTM. We know the particle receptive field that it was of the visual field, sorry, that it was sampling, and we know how it responds to transitions towards more luminance. That means white stimulus on a gray background, or to transitions to lower luminance. That means dark stimulus on a gray background as well. What you see in the bottom are the 2D maps of the receptive fields. In this case, what you see in blue, there is inhibition to the presentation of the white spot on the gray background that is confined to a circular region in the middle of the visual field there. And what you see on the right, in red, is excitation to the presentation of the dark spot on the gray background that you see even more confined space to this central circular region. This is roughly the shape of the receptive field. You have excitation at the very central spot to the dark spot and inhibition to the bright spot. And then you get that inhibition is wider in space. The blue circle is much wider than the red circle. And then you have the surround where the response is complementary. But let's concentrate on the center, because this is the data that we're going to use to investigate how the retina is wired up to the LTM. So the structure, the spatial structure of the excitatory center in red and the inhibitory center in blue is going to give us the clue of how many cells in the retina are wired up to this particular cell in the LTM. Now, we can describe this situation, this center, excitatory inhibitory center, in the large population of cells in the LTM that we have recorded from using 4 parameters. One is the size of the excitatory center in red, the other one is the size of the inhibitory center in blue, the other one is the relation between those sizes, and the other one is the eccentricity. As you can see, the center, the very center of the excitatory and the inhibitory domains is displaced in the space. Now, the first thing we thought was, okay, we think we can explain everything. We think we can explain why this asymmetries emerge in the way excitation and inhibition is represented in the center of the receptive fields. Imagine, if you imagine where information is coming from to these receptive fields, you can think of a circuit like this, like the one in okay, great. You can imagine a circuit like this, in which this is the cell that you are reporting from. All neurons in the retina belonging to the off mosaic and to the on mosaic are excitatory neurons, which means that excitation must come directly from a projection like this, in which this cell is receiving information from a cell that belongs to the mosaic of the same sign, but inhibition doesn't work. Well, inhibition must be routed through, because all these retinal cells are inhibitory, must be routed through these local interneurons in the thalamus that reverse sign. So the other mosaic, the on-center mosaic, is feeding onto interneurons information that reverse sign to create a inhibitory center in the relay cells. Okay, the question is how many of these cells actually contribute to each branch of the circuit. And in order to do so, what we did was just to come up with a computational model. Lucky us, we know a lot about the spatial distribution of units in the retina. This is something that we know since the 80s. And we know the properties of the receptive fields of each one of these units, in part because we have recorded from them and we have characterized them. And then the only thing that we have to do is to come up with a mosaic in the LTM that preserves the relative number of units of both signs, on-center and off-center, both excitatory and inhibitory units, that preserve the spatial distribution that we know they have in the LTM. And in order to wire them up, we came up with the simplest rule possible, which is a Gaussian rule that is represented by the equation below the mosaics. Mosaics are highlighted, or 2 sectors of the mosaics are highlighted below. And what that Gaussian rule tells you, or the constraint that it imposes, is that connections are local. And the number of units in the retinal mosaic that are going to contribute or converge onto each particular neuron of the LGN is determined by changing this Q value, which changes the magnitude of the Gaussian function. In this way, if Q is very, very low, then convergence is set to the simplest case scenario, which is a one-to-one convergence. Each neuron in the LGN receives from just a single neuron in the retina. And if I increase the value of Q, I increase the convergence ratio up to 12 to 1 or even higher. Then what I do is I take the receptive fields of the units that converge into a single LGN cell, and I sum up the receptive fields normalized by the same Gaussian function, because their contributions are going to be different if they are very close together in terms of visual space or if they are farther away. And I end up with the receptive field of my LGN neuron. I do this for the excitatory branch, for the inhibitory branch, and for the connection from the inhibitory neurons to the excitatory cells, and I end up with the circuit that best reproduces the experimental results, which is exactly what I wanted to have. So here, what you have is simply, I'm going to fast forward this, but you have in the bottom of the top row, you have the experimental receptive fields with the different values for the 4 different parameters that we consider, and in the bottom, you have the match in the experimental, in the computational model of those receptive fields. And the circuit that produces this is a circuit that has those values of connectivity. So each LGN cell receives an average of 3.2 different retinal ganglion cell inputs. Each interneuron receives a slightly larger value, 4.3, and then up to 6 interneurons converge into a single relay cell in the thalamus. So this is important because different designs of retinal thalamic circuits actually treat inflammation, visual inflammation, very differently. And I'm going to show you how that happens. So first, we know how this circuit is actually completed, how the algorithm, the decompression algorithm, is actually set up. You have a statistical wiring, it's random wiring, of a 3-to-1 with a 3-to-1 conversion ratio. What we want to know now is, what does this circuit add to the information that the retina is sending out to the cortex? Now, I'm going to just use a toy model for this. Imagine that you have a very reduced retina with just 3 retinal ganglion cells. Each one of them has a Gaussian receptive field that samples a region in visual space, these 3 receptive fields on the left. Now you have to wire that up to a receiving layer in the LGN that has double the number of units, 6 units in this case. In the simplest case scenario, when you have wired up 1-to-1, you end up with the same exact 3 receptive fields. The only thing is that you have doubled your message. So in a way, you have added security to the message that the retina is sending to the cortex, but that's it. You don't transform it. You don't transform that message any further. Now, if you allow convergence, and this convergence is random, what you end up is with something like this, in which you shuffle the receptive fields in the output, the output receptive fields of the retina in the receiving layer in the thalamus, and you end up with this highest variety of receptive fields, which are basically interpolated versions of the retinal receptive fields. Now, these receptive fields actually cover visual space more homogeneously, and they are more diverse. So one thing that we can do is quantify the diversity of receptive fields that you have in the thalamus for the different versions of the circuit. And we can come up with a diversity index, which this is the way of computing it that you came up with, but I'm just going to explain it like this. Imagine that you have 2 cells in the thalamus that are sitting on top of that green spot right there. The spots that are the bunch of spots on top represent the retinal mosaic of one animal. Imagine that you have two thalamic cells sitting on top of the green square that are sampling that mosaic. When they receive just one single connection from the retina, they are both receiving input from the green unit right there. And this is what you see in the numbers on the left. You see the number of inputs for neuron 1, which will appear in blue. The ones that are exclusive to that neuron and not common to both of them will appear in blue is 1. The number of inputs of neuron 2 is 1 as well. The number of common inputs is 1. And since those inputs are exactly the same, the diversity index is 0. What you're going to see here is the evolution of the diversity index as I increase the convergence from the retina to the LDM. And as you notice, to reach a local maximum, which is roughly at 3.2, which is the exact same number of retinophthalmic convergence that we got from feeding the model to the experimental data. So nice. So the model that the cat is actually implemented from the retina to the LGM is maximizing the diversity of receptive fields that we have in the LGM. Now, this maximization of diversity comes with an increase in coverage, as I showed you before. The receptive fields actually are distributed more efficiently through visual space. So one way to compute this is simply to take the very centers of the receptive fields, display them in 2 dimensions, and simply take the ratio between the pixels of the image that are covered by receptive fields versus the total number of pixels. And if you do that, you realize that you also reach a local maxima for the very same value that best matches the experimental results, a convergence ratio of 3.2. Now, you have more diverse receptive fields, you have more units, they cover visual space more homogeneously. Do they carry more information, or can they carry more information to the cortex? In other words, does your channel have a higher capacity with this particular and optimal retinophthalmic circuit? In order to answer that, we just use information theory just to make clear explanation of what it means. Imagine that you have your retina with the same 3 different units located in 2D space in this case. Now, if you stimulate it with a very tiny point of light, you get different responses when that stimulation happens in different places. Now, with low stimulation, you have a 0, 0, 0. No receptive field is actually active, so you send no information to the cortex. This is obviously a toy model, but bear with me for a while. Now, if I present the stimulus in the position, that particular position in visual space, what you have is that you light up one of the receptive fields and not the other 2, so you send a bit of information to the cortex, it's 1, 0, 0. If you move it slightly to the side, you still send the same information because the stimulus still falls only into that one receptive field. You move it over a little bit more, and still the same answer. And it's not until you move it over slightly more that you actually activate the receptive fields and your message to the cortex changes. So for a relatively large parameter space right there, the cortex is receiving the exact same information regardless of the stimulus changes that we have introduced. Now, imagine that you wire up your thalamus. If you do it in the very simplest case scenario in a one-to-one fashion, you are in the same situation. The only thing is that more units contribute to the message, but still the difference between the message in the 3 central situations, so there's no difference. So it's only until you get to the one at the far right that you send different messages to the cortex. While if you have the receptive fields actually being interpolated versions of the retina scrambled through visual space, you get a different message for each one of the situations. So you can quantify this using information theory, and when you do that with the real model, you get to a very similar solution. You get to a point where you reach a local maxima, which is roughly between 3 and 4. It seems that visual system is therefore maximizing the diversity of receptive fields in the thalamus to increase diversity, and also the capacity of the visual channel in that sense, information from the thalamus to the cortex. Now, is this useful in any way for visual processing? So, in other words, can I do better with the information that comes out directly from the thalamus than I do from the information that comes out from the retina, which is kind of counterintuitive in a way. So I'm much better off at discriminating visual stimuli with thalamic activity than I am with retinal activity. To do that, we use Bayesian decoding. And it's simply, the situation is just like this, that we stimulate the mosaic in the retina and the different versions of the thalamus wired up in the different ways, the 1-to-1 up to the 12-to-1 in convergence widening. And we ask, what is the error that we make when we take population activity in the retina and when we take population activity in the different versions of the thalamic circuit to estimate the location of the stimulus that we used as to evoke that population activity. So this is what you see here on the right. Should appear anytime soon, okay? And what you see there is, for different signal to noise levels, the dotted line represents the error in localizing the visual stimulus that you make taking the population activity in the retina, and the continuous lines is the error that you make when estimating position of the visual stimulus from the population activity of the LDM. And you see that except for the situation in which you are in a one-to-one fashion relative to the LGM, you are much better off taking the thalamic population activity. The error that you make is smaller than the error that you make using the retinal activity. And that is particularly true for especially for that signal to noise. That's particularly true for convergence ratios that are between 3 and 6, which is amazing that it also matches as well the experimental result. Now, to give a flavor of what it means in terms of seeing, how the cat would see if it only had the information coming out from the retina or the information that comes from our best thalamic model. Let's see if it works. Okay. What you see here is visual stimulus, which is in this case handwriting, is in blue. In magenta, you have the retinal prediction of the position of that stimulus, and in green, you have the thalamic position prediction. So at the end, they will separate, and you'll see. Oops. I'm going to let it finish, and you'll see how the differences that okay, so with the retina, you know, the many mistakes that you make, and sometimes in some parts of the text some parts are really difficult to read, while in the end, the situation is much more improved. The thing is that you have a second phase of upscaling of information, which is even larger from the LGM to the primary visual cortex. And this is something that we are investigating at the moment, so I'm not going to discuss it with you. But there's a dark side to increasing the resolution of an image through interpolation. Do you know which one is it? I mean, you must be very familiar with this. By doing interpolation, you're blurring the edges of your visual image. And this is something that also happens, not only happens when you take one small image like this and you blow it up. To increase its size, edges are actually blurred, extremely blurred. It also happens in biological vision. It's the same problem. We do have the same problem, but we don't see that way. We don't see that effect. There has to be something that counteracts these side effects of interpolation. And this is something that the inhibitory neurons in the nucleus actually deal with. This is the last part of my talk, and so it's very much smaller. So just bear with me for 5 more minutes, and that will be it. So I showed you that these cells, the excitatory neurons, actually respond in this push-pull fashion to visual stimulus. So if you have an on-center relay and you present a bright stimulus on a gray background, you get excitation followed by inhibition at the stimulus offset. And you get the complementary response if you stimulate the same place in visual space with the complementary stimulus. So if I present the dark spot, I get inhibition followed by rebound excitation at stimulus offset. This is below the equation that will explain this type of behavior. But in inhibitory neurons, you have the exact same thing. And here I'm showing you an inhibitory neuron that has the opposite sign. So it's an off-center interneuron. It gets excited by a dark spot on a gray background and inhibited by the bright spot on the gray background. Now, what we have also shown is that this type of complementary units, excitatory and inhibitory cells, form functional pairs, which we call them push-pull pairs, which process information in a very particular way. They imagine that by inhibiting each other, they allow the system to linearize responses to luminance and to increase the dynamic range of the system. So imagine that you have no inhibition in the system and you stimulate that on-center cell with luminance. What you end up with is the green curve in which for lower values of luminance you get nothing, and when you get to a threshold, then the response is roughly linear, increases linearly to luminance. In the interneuron, you would have the opposite, the largest response to the lowest luminance, and then it will decrease until you reach a threshold, you will get nothing. If you combine them both, you get the gray curve, which is a linearized version of the luminance, and it reproduces faithfully the transitions of luminance from 0 to 255. Now, we have shown that these cells actually get together in the nucleus. These form functional but also anatomical synaptic pairs. The funny thing is that they get so close together that the circuit saves 100 kilometers of wire in a very small region of the brain. It's a minimum wiring paradigm that gets these two types of units together. 11 kilometers of wire are saved in a very tiny region of the brain. So we're very confident that this is the exact circuit that is producing the transformation of the retinal image on its way to cortex. Now, what it does to the image is something very similar to what a morphological working filter would do. A morphological working filter takes, imagine that you have a stimulus which is a perfect step in luminance to go from something that is extreme, that is pure dark to something that is pure white in an instantaneous fashion. But if you have interpolation, you blur the edge and you get a red line. And then one way to overcome that is this artificial blurring is to use, for instance, an image working filter in which you set up some sort of a threshold and you take all the values that are smaller, which the threshold should be midway in the ramp of the red curve. And then you take all the values that are below that threshold and make them artificially zero, and all the values that are above that threshold and make them artificially the maximum, and then you recover the exact instantaneous step. The thing is, well, this type of filtering is very prone to noise and other artifacts, but what the brain does is something that is very smart in a way, and it takes the blurred version of the response, which is in red, and by adding pull, by adding inhibition, it actually stretches it up. So it does something very similar to the image working filter, but it does it locally, taking into account the amount of information on the on channel and the off channel there's locally. So it's not as prone to noise. So another thing is that you have on-center push-pull pairs and off-center push-pull pairs competing for the same region in visual space and inhibiting each other in such a way that you do this normalization of the image separately in different regions for the on-channel and for the off-channel. In some regions, the on-channel will dominate, in some regions, the off-channel will dominate. This is something that I'm working on with Eduardo Sanchez Villa, the University of Santiago here now at the moment. Now imagine what something like this would do to a visual image such as this. The histogram equalization that it would allow you to do is much more precise than the rough histogram equalization that I just presented before. It's something closer to this. And the image, the quality of the image is much better now compared to the original image. It's equalized, but it's equalized independently for the on-channel and the off-channel and at the local level. So this is basically what your brain's camera is doing all the time. It takes very small images with poor resolution, it blows them up on a first step of interpolation, and then using inhibition, it compensates for the undesired side effects of interpolation. Now, we are showing that this provides an increase in dynamic range of the image. It flattens the histogram. It has a very good local contrast enhancement performance without outputs or other visual artifacts. And also interestingly, we think that it explains other visual perception phenomena that are not easily explained using the traditional models, which is, for instance, simultaneous contrast or Mach bands. We can discuss later on about the Mach bands, but Mach bands, you see the artificial dark and bright bands on the ramp of luminance on top that are absent in the case of a very perfect and sharp edge. These Mach bands are not only an undesired side effect, in some cases are actually a desired side effect. In cases, for instance, in art, Greco used to use them actually to increase the saliency of visual borders in his paintings. And I just want to thank the people who did the work in my lab, particularly those two graduate students, Diego Alonso, who did most of the experiments, and Manuel Bolano, who did the computational model, and then my external collaborators, Fritz Sommers and Xin Wang, and lately Eduardo Sanchez Vila, who I had many helpful discussions with him, who contributed enormously to this work. So thank you very much.
Question. We are a little bit out of time.
Thank you very much for a very interesting talk. Neuroscience is always interesting. You touched on the push and pull models. I think it's fascinating. It explains why we have this kind of explained electric inhibition. But what's the downside? What's the downside?
I don't know. Very good question.
For example, if you see the consensus mass and people perceive some ghost triangles, do you think that's probably brought up by this?
There are many illusions that can be explained by a push-pull circuit, and that sometimes are undesired. It generates some sort of distortion in some images. But I think that that's an inconvenience that I would hardly call it a downside, because it actually is so useful most of the time that that's a small burden that you have to actually take.
Probably is not the right word, downside, unnatural, if I put it that way. Unnatural.
But I agree with you. I agree with you. Yeah, I agree with you. Thank you.
And also you touched on the face recognition. Do you think humans are particularly hardwired to recognize faces?
That is true. Faces are probably a particular case scenario. The thing is that for other objects in B4, we have receptive fields that can act as primitives. And these receptive fields are wired up in a very similar fashion to the one I just have described. This is something that was published 1 year ago as well. Meaning that the same type of mechanism which we represent luminance in a push-pull fashion here might actually set up competition between different features of higher complex objects. I'm not sure about faces, because faces are such a particular, as you said, such a particular case and unique to humans, basically, because of humans and higher primates. But I wouldn't be surprised if the selectivity to faces is set up in a similar fashion as well.
Thank you very much. We have 20 minutes coffee break.
Thank you, Professor Martinez.
Other rooms will be on the upper floor. After. Okay.